All right, so thinking back to last time, we finished up talking about the plasma membrane and the structural makeup of it, including phospholipids, which are by far and away the most numerous structures, along with various proteins. As you can see, we're going to talk a lot about some of those proteins like the channels and the carriers, because what we're going to be doing right away here now is talking about stuff moving in and out of the cell. What we call cellular transport. Okay, you can think of your plasma membrane kind of like a strainer that you use to make mac and cheese. I always use that as an example. We eat a lot of mac and cheese in my house. So I've got young kids. Plus, mac and cheese is delicious. Okay, if you're not familiar with how to make mac and cheese, I'll explain it to you like you're 2 years old. Boil water, you put the noodles in, let the noodles get soft, and then you take your boiling noodles and you dump it into the sink, into a strainer, colander, whatever you call it. That way the water goes down the drain and the cooked noodles collect in the strainer, okay? Well, your plasma membrane is like a strainer and then allows some things to go through it, but it blocks other things from traveling through it, okay? The term permeable means that a structure permits the passage of a substance through it. So in other words, you could say that a strainer is permeable to water. It means that water can pass through it, okay? While the term impermeable means that a structure does not permit the passage of a substance through it. So in other words, a strainer is impermeable to the movement of macaroni noodles, okay? That's why they don't go through, because the holes are too small, okay? Well, the permeability of plasma membrane to different substances varies. We call this property selective permeability, and a strainer also has this property, allows some things through, doesn't allow everything through. Okay, we can say that the lipid bilayer is highly permeable, so these things easily pass through, and there's a big fat purple arrow to represent this. Two nonpolar uncharged molecules, oxygen and carbon dioxide, and that's great because we want their movement to be really easily. Oxygen we breathe in must get into our lungs and then get into the blood and then leave our blood, go into cells. So it's got to go through a lot of plasma membranes. CO2 is going to go in the opposite direction so that we can eventually exhale it out. And then steroids are also very easily going to pass through. And that's because, and you can see illustrated here, the majority of the structures make up our plasma membrane are phospholipids, and phospholipids are a form of fat. So are steroids, and fats can generally move through other fats really easily. So fats like steroids are going to be easily passing through. So we'd say it's highly permeable. Okay. Lipid bilayer is impermeable. So these things will not pass through. And you can see we've got a U-turn arrow here showing that they're getting rejected to ions. So things like sodium, potassium, and chloride, as well as large uncharged polar molecules like glucose. But these structures, whether it's an ion or a glucose molecule, do need to come in or out of the cell, which is going to be done thanks to those channels and carriers that I introduced to you guys the other day, the doorways that allow substances to pass through the bilayer. And they do so in a very selective manner, and how, we talked about like sodium ions have travel through a sodium channel, chloride ions travel through a chloride channel, okay? We can say that the lipid bilayer is slightly permeable to small uncharged polar molecules, such as water and urea. And that's why you can see much smaller of an arrow. Water and urea are able to pass through, but for the most part, what we're going to operate around with water is it's more like an ion, and it kind of gets rejected, and so it's going to need a special type of protein channel, and we'll get into that here in a little bit. Now, the ways that substances move across and through the plasma membrane can be either through passive or active processes. Passive processes involve the movement of substances moving down their concentration or electrical gradient using only their own kinetic energy. Now, on the next page, we're going to talk about what a concentration gradient is, what an electrical gradient is. But if you notice, as I read that sentence, I really emphasized the word down their gradient, okay, down their concentration gradient, down their electrical gradient. I'll explain what that means here in just a second, okay? Active processes involve the movement of substances against their concentration or electrical gradient. And if you notice, I really emphasize the word against, okay, and I'll explain what that means in a second. Now, in active processes, cellular energy, usually in the form of ATP, is necessary to drive the substance uphill. So in other words, if we are dealing with any form of active transport or active process, we need ATP for it to occur. Whereas if it's passive, it does not require energy in the form of ATP. It's because it's got its own kinetic energy that drives it, okay? Now, there is another active process allowing substances to enter or exit a cell, and this is through small spherical membrane sacs called vesicles. We literally call it vesicular transport. It is an active transport process, but you don't really see ATP involved with it. We'll talk about it at the end of the lecture. Just know that it is another active process, but it's just kind of misleading because you don't really see the ATP involved. All right, let's talk about those gradients I just referred to. Okay, the selective permeability of the plasma membrane allows a living cell to maintain different concentration of certain substances on either side of the plasma membrane. In other words, and you can see an illustration of it here on the bottom of the page, okay, we represent in the kind of A box here on the left, maybe this is outside the cell, B over here, this is inside the cell, and then this line here where the letter I is represents the plasma membrane, okay? These numbers of whatever these blobs are, let's just say that they're oxygen, are not equal on both sides, okay? That's a situation that's very common in the body where you've got different numbers of something on one side compared to the other side of the plasma membrane. Sometimes it's more something inside the cell, sometimes it's more something outside the cell, okay? Now the movement of a molecule that has no net charge, or in other words, it's nonpolar, so that would be like oxygen and carbon dioxide, is determined by what we call its concentration gradient of the molecule across the membrane, okay? Well, basically what that means is we always determine the more common type of motion, what we call passive transport, based on this concentration gradient, the amount here versus here, okay? We always want to achieve equilibrium. And we've seen that word all the way back since the beginning of the semester. I kind of used that when I was talking about homeostasis. But if you don't like the word equilibrium, you can think of balance or equal, okay? So the way that substances typically move is through passive processes down their concentration gradient. So when I said on the previous page here that I'm back on, down their concentration gradient, it means I'm going from high concentration to low concentration, okay? And my advice would be to write that phrase out on that previous page. From high to low.
It doesn't require ATP.
And it doesn't require ATP. Okay. From high to low. So in other words, which one way? The natural progression is for substances, whether this is oxygen or water or whatever, is from A to B, okay? If we're going through passive transport, okay? But if we're going through active transport, we say it's going against their concentration gradient. So in that case, we're going from low to high, and I would write that phrase out. So with passive, down means from high to low, and with active, it's low to high. Think of it just like a ball in a hill, okay? On top of the hill, it's going to easily roll down from high to low, right? And it doesn't really require energy. But if you're pushing a ball up the hill, you have to continually push it all the way to the top because you're going against gravity, against the gradient, okay? And that's why we require ATP. So if we were talking about active transport, we would be going from B to A, from low to high, okay? So that's what we have for molecules that have no net charge. Oxygen, carbon dioxide, okay, are good examples, okay? But those aren't the only things that we have unequal numbers of. Our plasma membrane also creates a difference in distribution of positively and negatively charged ions between the two sides of the membrane. Typically, the inner surface will have more negatively charged ions and therefore is more negatively charged, whereas the outer surface is more positively charged. In our illustration, we show that this happens to be inside the cell, this happens to be outside the cell. And if you go through, you can see there's a lot of negative ions on the inside, not very many positives, whereas over here on the other side, there's a lot of positives and a few negatives, okay? This difference in electrical charge between the two regions, in other words, inside and outside the cell, constitutes what we call its electrical gradient. Okay? And another term for that is membrane potential, and we're going to get into that here in just a second, okay? Now, when ions move, because they can come into or out of the cell, thanks to those channels that we've been talking about, okay? This movement is going to be determined by what we call the electrochemical gradient. And that's the sum or combined effect of its concentration gradient and the electrical gradient, hence the name electrochemical. And this is just the gradient across the membrane, okay? So let's look at, for example, all these positive ions. We have a lot of them outside and very few of them inside, right? So if we were performing passive transport, and that means we're going from high to low, which direction would those ions be going? From right to left or left to right? Right to left, from high to low, okay? If you place an electrode inside and outside the cell, you're going to find that its resting membrane potential, which I'm getting ahead of myself, means that it's at rest, negatively charged. But what would happen if all those positive ions rushed in? Would that, whatever that number is, change, or would it stay the same? It would change, and we're going to talk about that right now. All right, membrane potential results from an excess of negative charge on one side of the plasma membrane and an excessive positive charge on the other side. There's a lot of negative on the inside and a lot more positive on the outside. Okay, cells at rest normally have a membrane potential, and I already referred to this, called the resting membrane potential, which is abbreviated V for voltage, sub M, and that's for membrane. Okay. You place an electrode inside, outside the cell, you can compare the ratio of negative to positive charges on the inside of the cell versus the outside, which is what this illustration did, okay? What you will find is that most cells, this is most, not every cell, has a resting membrane potential of negative 60 millivolts, okay? So it has a negative charge, okay? But that's just at rest. Okay, and that's just for most cells. What we'll find in our next lecture, which we're going to get into this, we probably won't get this far into the lecture, but neurons have a resting membrane potential of negative 70. Okay, but neurons and muscle cells exhibit what we call electrical excitability, the property of a cell to rapidly change its membrane potential as a result of a stimulus trigger. So in other words, a stimulus triggers this number to change. And it all is because of movement of ions. Okay, and I told you guys we talk about three main ions: potassium, sodium, and chloride. Okay, especially the first two that I listed, potassium and sodium. Okay, that's what we're going to be talking about in our next lecture, because that's what we're going to be talking about right there, action potentials, nerve signals. Okay, down here in our three figures on the bottom left, we have a graph representation on the distribution of these ions inside the cell, in the cytosol, versus in the extracellular fluid outside the cell. Okay, as you can see, potassium is very highly concentrated inside the cell, but very little of it is outside the cell. So the natural tendency is for potassium to travel from high to low, right? So going from inside the cell to outside the cell. And if that occurs, potassium rushes out. What would happen to this number? Would it become more negative, less negative, or would it stay the same? Okay, why do you say that?
Because it's going from a positive to negative. Or more or less. Is what I'm thinking.
Okay, so I'll just kind of explain it this way. This will just kind of simplify that. Potassium has a positive charge. Every single potassium ion exhibits +1, okay? Well, if potassium leaves, so do those positive charges. And if you lose positive charges, that's going to make this number become more negative. It's going to drop that number farther negative, okay? Whereas sodium in a plus, it's more highly concentrated outside the cell, so it wants to come in, and each sodium ion exhibits a +1 charge. So if a bunch of positive ions come in, guess what can happen to this number? It could become less negative, or if enough sodium comes in, we could actually become positive. We could travel past 0, which is what we'll find out actually does happen in our neurons, okay? Chloride is a lot like sodium in that it wants to come into the cell from the extracellular fluid, but does its movement make this number go more or less negative? It's going to make it become more negative, and that's because chloride ions are negatively charged. So that might cause us to become -90 millivolts. Whatever, that's just a random number. Okay. Does that make sense? Okay. Now, those are figures I actually snagged from a different textbook. I think it does a pretty good job at explaining and illustrating what's happening. These are the two figures from your textbook. They don't show anything for chloride, but it shows for sodium and potassium how they move from high to low, from outside in, which would make the resting membrane potential number change, becoming less negative, whereas if potassium rushes out down its gradient, it would make the inside of the plasma membrane more negative. Okay. And this will make a lot more sense as we roll through in our next lecture. So now that we have an idea of gradients, we can actually start talking about the ways in which substances move across the plasma membrane, either into the cell or out of the cell. The first method is through the process of diffusion. Okay, diffusion is a passive process. So right away, we know that since it's passive, we're going down the gradient, whether it's concentration or electrical. I'm not really going to worry about focusing on that. And since it's passive, we do not require ATP, okay? So it is a passive process in which the random mixing of particles in a solution occurs because of the particle's kinetic energy, not the ATP energy, it's the particle's kinetic energy, okay? And as we know, there's fluid inside the cell versus there's also fluid outside the cell. So this is all going to involve movement of stuff dissolved in water, right? Okay, so we're going to deal with solutes and solvents, okay? Both solutes and solvents. Solutes are the things dissolved. Water is the solvent that dissolves them, okay? Both the things dissolved and the water, the solvent, undergo diffusion. If a particular solute is present in high concentration in one area of a solution and low concentration in another area, the solute molecules will diffuse toward the area of lower concentration, meaning they move down their concentration gradient, okay, from high to low. After a period of time, particles become evenly distributed throughout the solution, and the solution is said to be in equilibrium or in balance, okay, or they're equal, okay. Easy demonstrations of diffusion can be done at home, okay. Take an air freshener or like a bottle of Febreze and push down on the button and spray it, okay? You're gonna smell that, whatever it is, floral, whatever scent, pretty much immediately, and it's gonna be fairly strong, okay? If I were to do that right here for you guys right now, Avery would smell it really quickly. It would take a little bit longer for you guys in the back to smell it, but eventually you would, okay? But over a few minutes, that smell would kind of dissipate, okay? And that's because all those particles have diffused around through the room, that they have kind of gotten diluted out. Now that's unless I completely empty the bottle, which I'm not going to do, but that would, you know, obviously saturate things a little bit more, and you probably smell it a lot more strongly, okay? Another easy way to do this is by using a glass of water and some food coloring, okay? Putting a drop of red food coloring in a glass of water, you're going to be able to literally watch diffusion taking place. And then eventually what's going to happen is all that red fluid will be dispersed throughout, and what you'll be left with is just a glass of almost kind of a real light pink, depending on how many drops of the food coloring you add. Okay. Now there's several factors that influence the diffusion rate of substances across the plasma membrane. The steeper the gradient, the faster the diffusion. Okay, so the greater the difference in concentration between the two sides of the membrane, the higher the rate of diffusion. So going back to this picture, if you have a heck of a lot on one side and very few on the other side, you can say that's a really steep gradient. Okay, and going back to kind of my hill analogy, the steeper a hill is, the faster the ball will roll, right? Okay. Well, if we have, like 8 and 7, that's a gradient, but there's only one difference. Okay. There's only a difference of 1. So that's a really, really, really not steep slope. Okay. It's almost a flat slope because it's almost not a slope at all. Okay. So in that case, it's not going to diffuse as rapidly. Okay. Now, when charged particles are diffusing, the steepness of the electrochemical gradient determines the diffusion rate across the membrane. So it's the same thing, just dealing with ions instead of something like oxygen that's not charged. The temperature, the higher the rate of temperature, the faster the rate of diffusion, which in our body is not really a huge issue because we're pretty warm. So that's fantastic. We get diffusion occurring pretty rapidly. Going back to the example of using like food coloring and water, if you take 2 glasses of water, one with hot water, one with cold water, and you drop food coloring in at the same time, you're going to see the diffusion occurring in the hot water a lot faster than it does in the cold water. And it's all because of the movement of the molecules in the hot water versus the cold water. I won't really get into that. The diffusing substance's size, the larger the mass of that diffusing particle, the slower its diffusion rate. Basically, we just think of it, if it's bigger, it moves slower. That's an easy way to remember it. A surface area, the larger the membrane surface area available for diffusion, the faster the diffusion rate. Remember the other day, we talked about microvilli, the little finger-like extensions, how they increase surface area, and that's important for places like our intestines where we have to absorb. Well, absorption is just diffusion taking place where something's coming into a cell, okay? So places like our small intestine allow for diffusion really, really rapidly to occur because there's a lot of surface area. Our lungs, another place that we have to diffuse, in this case, oxygen and carbon dioxide. They don't have cells with microvilli, but instead we have lots and lots of surface area because of structures called alveoli. These are like little tiny balloons inside of our lungs that actually do the gas exchange. We'll talk about that later in the semester. And then diffusion distance, the greater distance over which diffusion must occur, the longer it takes. Think of it, when you walk to your car, if your car is a mile away, it's going to take longer than if it's 2 feet away. Okay, farther you have to go, the longer it takes. The great thing is diffusion across the plasma membrane takes only a fraction of a second because our membranes are so thin. Okay, so that's not really an obstacle. Okay, so that's fantastic. All right, so that's just diffusion, but there's several forms of it. We'll start with the simplest. I guess that's why they call it simple diffusion, which is the way that a few small nonpolar molecules like oxygen and carbon dioxide move across the plasma membrane. We can describe simple diffusion as the direct, unaided movement of molecules into and through a lipid bilayer in the direction dictated by the difference in the concentration of the molecule on the two sides of the membrane. Okay, so in this case, we're always moving towards equilibrium, which means we're going from high to low, and it's an unaided movement. So it does not need the help of membrane transport proteins. So we don't need channels and carriers for oxygen and carbon dioxide to come into the cell. This is like in movies when you see ghosts floating through a wall. Okay, they can go directly into the room without using a doorway. Okay, in the case of oxygen and carbon dioxide, that's fantastic. They can do that. So can nitrogen gases, fatty acids, steroids, and fat soluble vitamins. Remember, fatty acids are fats, steroids are fats, fat soluble vitamins are fats. What did I say earlier? Fats can travel through fats, right? Okay, so they don't require anything to go across the plasma membrane. They are like Casper the Ghost, floating from high concentration to low concentration. But for most substances, which include ions and things like glucose, we need a transport protein to help us, or to help it, I should say, to help them move. And that process, using a transport protein, is called facilitated diffusion. The word facilitate, just kind of a fancy word for help. So this is just diffusion going from high to low with help. Okay, who's helping? A transport protein, either a channel or a carrier. Okay, and so when a transport protein moves a molecule down its free energy gradient, concentration, electrical, or both, we call this facilitated diffusion or passive transport because it requires no energy. So once again, we're going from high to low, no ATPs needed. Difference between the two is simple. If it's channel mediated, we're using a channel. Remember, that's where the protein doesn't change its physical shape. If we're using a carrier, it's carrier mediated. Okay, and that's the type of protein that does physically change its shape. Okay. Now, in the case of channel mediated facilitated diffusion, typically it's moving ions through ion channels. So this is a passage of small inorganic ions that are too hydrophilic to penetrate the nonpolar interior of the lipid bilayer. Remember, the tails of those phospholipids are hydrophobic, and so they don't like water and other charged particles. Ions are charged particles, so the tails don't like them, and that's why they have to use channels. Okay, and then we talked about the other day how channels can be gated or have like a door that covers them when part of the protein acts as a plug or a gate, changing the shape in one way to open the pore and another way to close it. In our next lecture, we're going to talk a lot about ligand gated channels, which open up when a chemical binds to that channel. We'll talk about voltage gated channels that open up when a specific voltage on the inside is met. And we'll get into all that in our nervous tissue lecture. Facilitated diffusion is a very fast process through a channel. It, for example, allows for more than 1 million potassium ions to flow through a singular potassium channel in 1 second. So very significant. Once again, carrier-mediated diffusion involves a carrier moving a solute down its concentration gradient. In this process, the protein must change its physical shape, so the solute binds to the carrier on one side of the membrane, released on the other side after that change has occurred to the carrier protein. Here we can see glucose binding, and I can tell you this is glucose just because I know the shape. But the pore is open on the outside, and then what's going to happen is a physical change occurs to the protein and spits it out, in the case of this, into the cytosol. Whereas over here with our channel, this is an ion, whatever it is, it's just going to go directly through and not change the protein shape. In the case of carrier-mediated, the rate at which this occurs is determined by the steepness of the gradient across the membrane. So once again, the difference between the two sides, the greater the difference, the steeper the hill, the faster the rate of diffusion. Make sense? Okay, there is one final form of diffusion, and we call this osmosis. Osmosis is diffusion specifically of water through a selectively permeable membrane, or in other words, through the plasma membrane. Okay, and this is possible by way of what we call aquaporins, fancy term for our water channels. So the channels that move water through the bilayer from one side to the other. In order for this to occur, there must be a concentration difference across the membrane, which results in the solution on one side of the membrane having a higher concentration of solutes and thus a lower concentration of water on the other side. Okay. Osmosis also needs a membrane more permeable to water than to the solutes. Okay. In the case of osmosis, it's just we're moving from areas of high water concentration to the area of low water concentration, which is just down its concentration gradient. Okay. Now I've got an illustration. This is from your textbook to kind of show you how osmosis works. But I actually really like the figure that's on the next page a little bit more. Okay. And so I'll talk about it and I'll kind of back to that, or go back to that page. Looking at this illustration, we've got solution 1, which is a solution with 5% albumin. Albumin's a protein, whereas solution 2 has 10% albumin, a lot more albumin. Now, looking at just the figure without reading anything, you probably would assume that this jar is solution 1, and the one on the right would be solution 2. That's not actually the case. Okay, it's the same jar, just here is at the beginning of the experiment, and then after time, doesn't really matter how long that is. This is the same jar, and what's happened is a change. Okay, so what solution is, or solution one is this side of the jar. Solution two is on the other side of the jar, and they're separated by a membrane, like our plasma membrane, that is permeable to water, so water can pass through. But albumin, it's not permeable to, it's impermeable to albumin. So albumin, which are the yellow dots, cannot pass through. Okay. Now, looking at the jar here at the beginning of our experiment, we would say that these two solutions have an equal volume. There's the same amount, right? Okay. What we end up with is not that way. But would you say that solution one and solution two have the same amount of water? The answer should be no. Okay. If this was rewritten, instead of saying 5% albumin, 10% albumin, if it read something like this, that might make a little more sense. Let's say it's not 5% albumin, but 95% water. And then we'd say this is 90% water. So now would you say there's the exact same amount of water? No, solution one has more water because it's got less other stuff. Solution two has less water, but it's got more other stuff. So this is our high water concentration. This is our low water concentration. And osmosis goes from high to low. That's why you see the big blue arrow, much more than the arrow going the other way. Okay, so more water moves this direction versus this direction, which is why solution one, its volume drops. Solution two volume goes up. And what we're left with, and as a result of the experiments, we have reached equilibrium, where both solutions now have the same amount of solutes, 7.5% and 7.5%. Okay, so we can say that we go from high water to low water concentration, but if you reverse it, you could also say osmosis goes from low solutes to high solutes. Low other stuff to high other stuff. If you think of it that way, you can write it that way, but this is another kind of way probably to confuse yourself. Now, when osmosis occurs, one solution loses volume and one gains volume, which we show here occurring just the same way, except we started with nothing else. Versus over here, there's a lot of other stuff in there. We don't really care what that solute is. One volume goes down, the other one goes up. But in our two examples, this one and the one on the next page, we see an open-ended tube or an open-ended jar. Okay. Well, cells are not open-ended. They are closed compartments. So they are demonstrating here by pushing in with a piston what it's like inside a cell when that water rushes in. The change in volume is always going to be accompanied by a change in pressure. Okay. Think of a cell like a water balloon. The more water filling up a water balloon, the more pressure inside. Okay. Okay. So water pressure that develops as a result of osmosis into a solution is called osmotic pressure. Does that make sense? And that's important because of what we call tonicity, which I'll talk about now. As water moves by osmosis into or out of a cell, changes in volume either occur, either increasing or decreasing. Okay. A solution's tonicity is a measure of a solution's ability to change the volume of the cells by altering its water content. Okay. So in other words, when we talk about a solution, we can describe it as isotonic, hypotonic, or hypertonic, referring to what it's going to do to the inside of the cell. Okay. An isotonic solution can be described as having the same potential osmotic pressure because they have the same concentration of impermeable molecules. which results in water moving in and out at the same rate. And that's what we illustrate with this figure on the bottom left. So what this solution is referring to, which solution we're talking, isotonic, hypertonic, hypotonic, whatever, it's the fluid in the jar, okay? So it's this water plus whatever else, okay? If we just want to be simplified, let's just say it's water and salt, okay? In this case, we say an isotonic solution has the same amount of water and salt. Maybe it's like 95% water, 5% salt, okay, just to put some numbers on it. Here, well, that's the exact same thing. That's what's inside the cell. So there's no high water, low water. It's equal already. So there is no high to low. It's already equalized. So stuff comes in and out at the same rate, okay? And that's great because that's not going to screw with the shape of a cell, okay? Like you see here in our figure on the right of a red blood cell, okay? In a hypotonic solution, hypo is a prefix that means low, and a hypotonic solution, there's a lower level of solutes than in a living cell, causing a cell eventually to swell, I should say to swell and burst, a process known as lysis, okay? So in a hypotonic solution, this is like just having pure water. So this is like having 100% water and no salt, okay? And it's gonna rush that water into the cell because the cell has some salt, okay? So we have high to low occurring to the point where if so much water comes in, the cell can actually burst. And if this happens to a red blood cell, it's called hemolysis. And this can actually happen if a person drinks enough water. People do die from drinking too much water. I mean, you have to drink and drink and drink and drink and drink, okay? But it does happen, okay? A hypertonic solution is the exact opposite of a hypotonic solution. A hypertonic solution, hyper means high, so there's a high level of solutes and a higher level of solutes than a living cell, causing water to move out of the cell, okay? So this is like having salt water from the ocean, okay? A lot of salt, okay? A lot of salt here sucks the water out of the cells to try to balance that out, okay? Causes the cell to shrink, which is called crenation. Actually, you can see it really changes, screws with the shape of the cell, okay? So this causes water to flow out, okay? This is why if you're stuck in a life preserver at sea, what are you not supposed to do? Drink the ocean water, because what does it do to your body? It makes you dehydrate. This is what happens, okay? All that salt in the water ends up in your bloodstream, and so your body tries to dilute that salt by taking all the water from our cells everywhere else and puts it into our blood. And all those cells lose their water, so they're dehydrated. Okay? It makes you go crazy, hallucinate, and then it can cause you to die. Okay? So a hypertonic solution causes water to move out of the cell. Doesn't say it directly, so I would write this out. Hypotonic solution makes the water flow in. So hyper out, hypo in. Isotonic is just at the same rate. Okay. Now, I know a lot of you guys are planning on becoming nurses, and one of your jobs as a nurse will be to administer IVs. So let's talk about that. Our red blood cells and other body cells may be damaged or destroyed if exposed to hypertonic or hypotonic solutions. For this reason, most intravenous IV solutions are isotonic. Examples are saline, 0.9% sodium chloride, and D5W, standing for dextrose 5% and water. Okay. That doesn't mean that sometimes a hyper or hypotonic solution isn't beneficial to the patient. Infusing a hypertonic solution like mannitol, sugar alcohol, is useful to treat patients with cerebral edema, which is excess interstitial fluid in the brain. In other words, they got swelling on their brain. And so that water that's around their brain needs to be eliminated. So infusing that solution causes the movement of water from the interstitial fluid into the blood, and then the kidneys take that water and make it into urine to be peed out. And that will help with that cerebral edema. In the other case, what we call a hypotonic solution, it can be given orally or through IV to treat people who are dehydrated. Okay, the water in the hypotonic solution moves from the blood into the interstitial fluid and then into the body cells, just like we show here, hopefully not to the point where they explode. This is what water, just water that we drink and most sports drinks that we consume to rehydrate after a workout are relative to your body cells. Make sense? Okay, so those are the different methods of passive transport. What we're going to do now is talk active transport, which is the way that some polar or charged solutes must enter or leave the cells. And in this case, we're moving uphill, which means we're going against the concentration gradient. In other words, from low to high, okay? And when a transport protein moves a molecule against their free energy gradient in this process, we call it active transport because it requires energy in the form of ATP. Two forms of active transport in the human body exist, primary and secondary. There is the third that we'll get into in a second called vesicular. First, let's talk primary and secondary. The type of transport or active transport that we talk about the most in this class is primary active transport. We call it primary because we're directly using the hydrolysis of ATP to power this process, okay? It involves a type of carrier protein called a pump because we pump substances against a concentration gradient against its, that doesn't make sense, cross a group. Just cross off across a concentration gradient. If so, it should read because they pump substances against its concentration gradient. That would make a lot more sense. Sorry about that. Okay. So anytime you hear me say pump or something's being pumped, you know we're talking active transport. So we're going from low to high and we're requiring ATP. Okay. Now, the most prominent example of primary active transport involves what's called the sodium potassium pump. Okay. So you'll also see it listed as sodium potassium ATPase pump. Same thing. Okay. This is where we transport 3 sodium ions out of cells, 2 potassium ions into cells. Okay. Which is against both of their gradients. Let's just look at our illustration at the bottom. We show in blue at the top of our figure the extracellular fluid, which, as you can see, has a lot of sodium and not very much potassium, okay? Whereas inside the cell, on the cytosol, there's very little sodium and there's very high potassium. So the normal progression is for sodium to want to go into the cell and potassium to go out of the cell, just like we talked about earlier today. But we do the opposite of that, okay? It's a 4-step process. These are not 4 different carriers. It's the same one, okay? It's just 4 different points in time. Okay, here where the pores open to the center of the cell, the 3 sodium ions can bind to 3 special spots, okay? And then using the energy harvested from ATP, we expel those 3 sodiums. We go from low sodium to high sodium, okay? And then that exposes the pore to the outside where we can now attach 2 potassiums, and we then change the protein shape again to import the 2 potassiums from low to high. Okay. So for both sodium and potassium, they're moving against their concentration gradient from high to low, or excuse me, from low to high, and thus it requires ATP. Okay. Make sense? Now, this happens, which then helps drive this, what we call secondary active transport. And I'll explain what I mean by that as soon as you're done writing. In secondary active transport, the energy stored in an ionic electrochemical gradient is used to drive solutes against concentration, across the membrane against their concentration or electrochemical gradients. Because a sodium or a hydrogen gradient is established by primary active transport, secondary active transport indirectly uses energy obtained from the hydrolysis of ATP. Okay, Most secondary active transport systems are driven by sodium electrochemical gradient. In secondary active transport, a carrier protein simultaneously combines sodium and another substance and then changes shape, allowing both substances across the membrane at the same time. We have what we call symporters and antiporters, and I'll get to that. But first, let me kind of explain what I just read out because that was a lot of gibberish. Okay. So what we can do, if I go back to this previous page, kind of put some numbers and thoughts to what we talked about. Okay. So let's just say before one round of this sodium potassium pump, we had, let's say, 50 sodium ions outside the cell, and we'll use 20 on the inside of the cell, okay, which is a difference of 30, okay, because 50 minus 20 is 30, right? Okay, well, if this occurs once, where we now are moving 3 sodiums against the gradient, we go from having 50 outside, we gain 3, so we're at 53, okay? And since we lost 3 from here, now we're at 17. Okay, is everybody following along with my numbers? Okay, well, what did we say about the steepness of a gradient? The steeper it is, the more it drives diffusion, right? Okay, well, before this, our difference was 30. But now, after this occurs, 53 minus 17 is 36. So we went from having a 30 ion difference to now 36 ion difference. Has our gradient gotten steeper or not? It has gotten steeper, okay, because we have a bigger difference now than we did before. There's more, even more here now than there was before, and there's even less here, okay? So there's now an even bigger want to get sodium into the cell, okay? And that is what powers this process, okay? Sodium really wants to come into the cell, and so it's going to do that using either a symporter or an antiporter, okay? A symporter is a transporter that moves 2 substances in the same physical direction across the membrane, okay? So in other words, sodium gets to give somebody a piggyback ride. And sodium comes in and glucose takes a ride on its back. Sodium comes in and amino acid comes in on its back, okay? Well, look where the natural tendency for glucose and amino acids is. It's to go from high to low, from inside out. But instead, it's going against its gradient. So that's why this is active transport. Do you see ATP in this process? No, because it's indirectly using it. Because this happens first, that then allows this to happen. Now, the same thing happens in an antiporter. 2 substances are moving across, except they're moving in opposite physical directions. So in the case of sodium, it's still going to go from high to low through a protein. This time the protein is called an antiporter that allows sodium to come in, but something else to escape in the opposite direction. Calcium to go from the inside out. Hydrogen ions to go from the inside out. Both of those are occurring against their electrochemical gradient. Okay. So this happens after primary. That's what they call it secondary. It's kind of misleading because it's not showing you anywhere here that ATP is needed, but ATP was needed back here. So without primary, we can't have secondary. So without ATP, this doesn't occur. Does that make sense? We don't talk a whole lot about secondary active transport in our body in this class anyway, but it does happen. Okay. We will refer to it kind of probably 1 or 2 times, not very much. Okay, the third and final form of active transport is called vesicular transport. Okay. This is the movement of materials into or out of the cell via small spherical membrane vesicles. Okay, and it's a lot like what we saw the other day when we talked about the rough endoplasmic reticulum making proteins that then get carried to the Golgi with these little bubbles. Okay, except now we're either moving something into the cell or out of the cell. Okay, or a combination of the two. endocytosis. You say that, I always just like to pronounce it like endocytosis. Okay, this is where substances are coming into a cell in a vesicle formed by the plasma membrane. Okay, so it's exactly what it looks like on the next page. Okay, whatever these little purple dots are, it doesn't really matter, but the cell kind of just engulfs them. The plasma membrane pinches inward, and eventually it surrounds it, and a bubbles form called a vesicle. Okay, that's what endocytosis is. Now, there's three forms of endocytosis: receptor-mediated phagocytosis, bulk phase endocytosis, or pinocytosis. We talk about the second one there the most, okay, but I'll read through all three of them. Receptor-mediated is where it's a highly selective type of endocytosis by which cells take up specific molecules called ligands after binding to specific receptors on the plasma membrane. So it involves receptor binding to the cell, and then the cell knows, okay, I need to pull that in, and that's what happens. Okay. In the case of phagocytosis, this is cell eating. Okay. And this is what our macrophages do. The cells that I always tell students, think of them like Pac-Man, running around and gobbling stuff up. Okay. This is the engulfing of large solid particles like worn out cells, whole bacteria, or viruses. And actually, we see it taking place in some real life situations. A microbe, what this is, doesn't matter. Maybe it's a bacteria. Here's our white blood cell. It's engulfing it. It's coming in. It's like literally swallowing it up. Okay, and that's what this is showing you. That way, that microbe can be destroyed, because as we know, there are those garbage disposals of our cells called lysosomes with their enzymes that can eventually digest them. Okay. And then bulk phase endocytosis, which is also going to go by pinocytosis, is like cell drinking. It's going to involve tiny droplets of extracellular fluid being taken up, and a difference between it and receptor mediated is no receptor proteins are involved. Okay. The opposite of endocytosis is exocytosis. So think of this. We said this was endocytosis. Think of this like exit cytosis. This is where stuff exits the cell, where we fuse the vesicle with the plasma membrane, releasing the contents out of the cell into the extracellular fluid. Okay. So it's just the reverse of endocytosis. Now, this illustration is actually showing you what's called transcytosis. And that's just a combination of endo and exo, where the vesicles undergo endocytosis on one side, then that vesicle moves across the cell and undergoes exocytosis on the opposite side. So it's where we bring something in through endo, we then move that bubble across, and then on the other side, we spit it out through exocytosis. If you remember, trans is a prefix that means across or through. So that's where this term comes from. We don't talk a lot about this type of motion. Really, mainly what we talk about for vesicle transport is exo and endo. But this does occur in blood vessels to move materials between blood plasma and interstitial fluid. Okay. And like I said earlier, it's kind of misleading because we don't really show it in the figures that ATP is being utilized, but both exo and endo, and therefore trans, since it involves both of those processes. So both of them require ATP, which is why this transport is classified as an active process. All right, just to kind of summarize and simplify things, table 3.1 listed here, just lists off all those different forms of motion. We talked about each one of them, and gives a nice description and shows you substances that are involved. But I'm not going to ask you to directly study it or anything like that. Just included it because it was in your exam or in your textbook. All right. Last thing I want to talk about, and I'm just going to really briefly talk about it. I could spend an entire lecture talking about this, is what we call the cell cycle. And this involves a process called cell division, which we've kind of referred to a few different times. The process by which cells reproduce themselves. In other words, going from one cell to another. And what we're left with are two cells. Okay. Simple illustration of that is shown at the bottom. Not all cells of the body undergo cell division, but most of them do. Okay. And cell division is one of the components taking place in the cell cycle. The cell cycle is defined as an orderly sequence of events by which a typical or somatic cell duplicates its contents and divides in two. Okay. The cell cycle involves two primary phases. They are color coded here in our picture, the majority of a cell's life is spent in what we call interphase. Okay, this is shown here in purple. It's a kind of three-step process: G1, S, and G2 phases. Okay, but this is just the normal point in time where a cell is doing its normal functioning. Okay, whatever that cell does, it's doing its normal job. Okay, but something very important takes place during this process, what we call DNA replication. And that's what is being illustrated here in this picture, where we're duplicating the DNA. Remember, DNA is a double helix, and what we have to do with it is basically unzip it. So if you think of like a zipper, and then each of those original strands, since it's double stranded, there's two of those old original strands, and then we put a new complementary strand to it. And what we're going to be left with are two identical, hopefully anyway, double helixes of DNA. Okay, that way, when our cells divide and we go from one cell to two cells are going to hopefully match each other with duplicated and matching DNA. Okay, the actual point in time when the cell does this is through the S phase, and the S is actually short for synthesis phase. This is the phase where DNA is synthesized, it's replicated. Okay, now the red shade, I guess that's red shaded portion of our kind of pie chart there is showing you what we call the mitotic phase, what we just literally call mitosis, a process where the nucleus divides and then the cytoplasm also divides, which is called cytokinesis, and it involves prophase, metaphase, anaphase, telophase, the correct order, and then eventually at the end of that, we've got our two individual cells. Okay, if you can remember PMAT, which is how I was taught to kind of remember it. Okay, it's an easy way to remember the order. It involves some very sophisticated stuff taking place. Okay, this is a very, very simplified explanation of what's taking place during these different four phases. Okay, so keep in mind, like I said, I could talk much greater detail about it, but I'm simplifying things. This is all you need to know for this class. If this was biology, I get into much more detail. But the first phase of mitosis is called prophase, where we first see the appearance of chromosomes. If we look at the top of our figure on the right, here's our cell, but it's in interphase. And as we learned, most of the time, a cell inside its nucleus doesn't have its DNA in chromosomal form. Instead, it's got the DNA in the form of chromatin, which is that grainy mass. But when we get into prophase, we see those things that look like X's or H's, whatever letter you think. To me, it looks like an X. Okay, those are the chromosomes. And as we know, typically, how many chromosomes will we have? 46. Very good. Okay, we only show 4 here. So this is a very simplified illustration of it. Okay, but this is where we first see chromosomes appearing. Okay, we describe those chromosomes as appearing as sister chromatids. Okay, this X shape is actually two different, basically sideways V's. Okay, and so let me just kind of illustrate it. Okay, just draw real quick. Instead of thinking as an X, if I were to draw it more like that, okay, and if they were connected, I would, connect them. But these two things are sister chromatids, and they're identical to one another because this and this are the duplicated forms of DNA. Okay. Well, I go back to my figure. Okay. We see those are labeled. Okay. This is the first time we see those sister chromatids. Okay. During late prophase, we see that the nuclear envelope that surrounds the nucleus kind of disappears. It starts to break up, and we see those chromosomes kind of starting to organize toward the center of the cell. But really, when they're down the center of the cell is during metaphase. Okay, I always think middle for metaphase, and that's where they've aligned themselves literally around what's called the metaphase plate. Okay, Those things I referred to earlier in the lecture that look like spider legs are starting to come to the center of the cell and grabbing onto that center of those chromosomes, okay? That way, during anaphase, they get pulled apart, okay? Now those sister chromatids have broken free from one another and are being pulled to opposite ends. That's what happens in anaphase, okay? And then telophase is where we start to reform the two new cells. So the chromosomes will start to kind of go away. By the time telophase is over, we are left with that chromatin material again. So the chromosomes have gone away. The nuclear envelope starts to form. Those spindles, those microtubule spider legs start to disappear. And we start to see it more so probably in late anaphase, but definitely in telophase, what's called a cleavage furrow. And that's the first sign of cytokinesis, which is the first time when the cells are literally starting to show that they're breaking free from one another. And we're going to end up with two, hopefully, identical cells. Make sense? Okay, let me show you, because it's always great to show visualizations when possible.
If you're a college student like me, especially, you need Grammarly in your life. Grammarly is a digital writing assistant and.
Okay, so this is a real cell undergoing mitosis. You see, this is all the nucleus right here. You can see we're starting to see those chromosomes. All right, here we go. Here's where the motion is going to occur. This is all prophase still. All right, now we're getting ready for metaphase. You can see how they're starting to align towards the center. Okay. Get ready. Get ready. Anytime now. There they go. All right, so see how they're breaking free, pulling to the opposite sides. Now this is anaphase, okay? Now we're starting to pull the cells apart. Here we can see this is the cleavage furrow. This is where the cytoplasm is starting to divide. We've got two distinctive cells, okay? And you can see down here especially, those chromosomes are starting to uncoil. So we know we're now in telophase. And by the end of it, boom, we've broken the two cells free. And now we've got two identical cells. Pretty amazing. Now, unfortunately, associated with cell division is that.
As this plant cell, taken from a lily, prepares for division, the chromosomes first conduct.
Shush. Condition known as cancer. Okay. Cancer can be described as a group of diseases characterized by uncontrollable or abnormal cell division. So in other words, the body just starts dividing all these cells, and what ends up forming is an excess of tissue known as a tumor. Okay. Fancy term for a tumor is a neoplasm. And there's two types of tumors, cancerous and benign. Cancerous tumors are described as malignant, and malignant is a term that means they have the ability to metastasize. Metastasis is just a fancy way to describe that the fact that cells can spread from or an original site to somewhere else and a new tumor can develop. Okay, so cancer cell breaks free and then it gets lodged somewhere else. Okay? And then a new tumor can form, and that can be very dangerous. Now, not all cancerous tumors metastasize. Okay, some tumors, even if they're cancerous, don't always spread. Okay, but you know, like breast cancer, that's one that really metastasizes. That's why it's such a dangerous cancer. And other type of cancer or other type of tumor, excuse me, are benign. And I was taught to remember, if you know you're benign, you're fine. That rhymes. They're neoplasms that don't metastasize. So they're often considered to be non-cancerous. Does it necessarily mean that you're 100% fine? No, because benign tumors can still be harmful to the body. Okay. People can get benign brain tumors, and they can really interfere with normal brain function. Okay, but typically benign is the type you would want if you have a tumor. Okay. Now, cells of malignant tumors duplicate rapidly and continuously. As their cells invade surrounding tissues, they often trigger angiogenesis, which is the growth of new blood vessels. Okay. What's unfortunate about tumors and cancer cells, your body doesn't really know that they're bad. Okay. In some cases, they assume that they're just normal, good cells. And when growth of tissue occurs, growth of blood vessels to supply those cells occurs. Okay. So this can allow for growth of new tumors that may compete with normal tissue for space and nutrients, which often leads to death of normal tissue. Okay. So as tumors get bigger, they hog all the nutrients, they interfere with normal function. That's what ends up causing organs to shut down, and obviously can lead to death of the individual. Okay. There's a lot of really cool research on new methods to treat tumors and cancer. One of the coolest I've seen is where they actually will inject viruses into them. I can't remember. It was one of the that you typically, I think it's like smallpox, where they actually will inject it, and that activates the immune cells to alert them that your body naturally goes after those smallpox viruses. So it acts as a way to highlight to your immune cells that you've got these cells that are not normal, and it helps hopefully destroy them and allows your immune system to kind of kill off that cancer on its own. Something that might be a lot more beneficial than chemotherapy, which can help kill cancer, but also can be very debilitating to the rest of the body. So hopefully a lot more research is yet to come, because cancer is really, really unfortunate. All right, any questions?